Hey guys, so here we are, sections 8.1, 8.2, and 8.4, which is classification and the tree of life. We have three key concepts for this section. Organisms can be classified based on their physical similarities. Modern classification is based on evolutionary relationships. And the third one, the current tree of life, has three domains, and we're going to talk about each of these in just a moment. Our new vocabulary, we've got classification, taxonomy, taxon, binomial nomenclature, genus, phylogeny, cladistics, cladogram, clade, derived character, nodes, archaea, bacteria, and eukarya. Only one essential question, just how are living things classified into the tree of life? So if we look at this, every day you have to organize things, and you do it very, very quickly, and you don't even realize you're doing it. So if I was to give you these organisms here, how would you group them? I'm sure you've taken a few times. Maybe you can group them one way. What's another way you could group them? Did you do it like that? Did you put the blue ones together, the red ones together, the one that's red and blue kind of in the middle? You could have done it like this: fish, butterflies, birds. Could have also done like flying animals and swimming animals. So there's multiple ways you could classify these different organisms. And again, we do this every day with all sorts of different groupings of things without even realizing it. If I was to give you these organisms, how would you group them? Hopefully you're saying cats and dogs, because they are cats and dogs. Okay. All right. So anyway, to get into what we're going to talk about today, there was a scientist that we brought up when we talked about Charles Darwin by the name of Linnaeus, and he developed the scientific naming system we still use today. And he was a naturalist. He collected a ton of different specimens, and he looked at them and looked at the way they were put together, their structures, and organized them into different groups, just basically like he did with the birds and the fish and the butterflies. And so classification is the arrangement of organisms into orderly groups based on their similarities, just like what you did. Taxonomy is the science of naming and classifying organisms, and each little group that we make is called a taxon. So a taxon is a group of organisms in a classification system. Now, as scientists, when we talk about different living things, we use a scientific name. And so binomial nomenclature is a two-part scientific naming system. It uses Latin words, and the scientific names are always written in italics. And so the two parts of the name are the genus and then a thing called the species descriptor. The genus includes one or more physically similar species. And species in the same genus are thought to be closely related. And when we're talking about the scientific name, the genus part, which is the first part of the name, is always capitalized. The species descriptor is the second part of the name. It's always written in lowercase, and it always follows the genus name. It's never written alone. So if you look at this little barn owl that I have over there, his scientific name is Tyto alba. That first part, Tyto, you can see the capital T, that's his genus, so that T needs to be capital. His second part is species descriptor is alba, it's lowercase. Both parts of the name are written in italics, that's important. Okay. Now, we may have many different owls that belong in that genus of Tyto. They all belong to that same group, but only one specific kind of owl, the species, will have that name alba. So if we look at this, this is just a little graphic showing you scientific names. I've got Puma concolor. The genus is Puma, again, capital letter. The species, all lowercase, that's concolor. Everything is in italics. A common question on the EOC is to say which one of these is a proper scientific name. You're looking for a capital letter on the genus. Is it in italics? Is it including both parts of the name, the genus and the species? And again, we use a scientific name because many organisms have very many different common names. So if we might want to call this Puma concolor, we might call it a puma. We might call it a mountain lion. We might call it a cougar. It's all the same animal. So as a scientist, you want to be sure you are talking about a specific thing. So if we use a scientific name, there's only one organism that we can call Puma concolor. Okay? So if we look at these three species here, we have a giant panda, a polar bear, and a grizzly bear. Which two would you guess are more closely related? Look at their scientific names. If you guessed Ursus, so the polar bear and the grizzly bear, if you look at their genus, it's Ursus. Those two are more closely related. They belong to the same genus. They have different species, but they still belong to the same bigger grouping of genus. The giant panda, nowhere near that. It's in a separate group. Okay, some benefits of classifying and using binomial nomenclature. Well, it allows scientists to help them communicate. Many species have very similar common names, and common names are different all over the world. Like I might call something like that giant danger guppy, I could call it a giant danger guppy if I wanted to, but some other people might say that's a shark, okay? But I don't know specifically what kind of shark we're talking about, nothing like that, unless I was to use a scientific name. The Linnaeus classification has 7 levels, okay? So his naming system or his classification system involved 7 levels with kingdom, phylum, class, order, family, genus, species, and you can see how each grouping gets more narrow and more narrow and more narrow all the way down to species. Nowadays we have an extra level above it, which we'll talk about on Thursday. Okay, the Linnaean classification system does have limitations. It did not at his time count for molecular evidence because that technology wasn't around during that time. His system is only based on physical similarities, what he could actually look at and determine and then group things together. But as we know, physical similarities are not always a result of close relationships. Genetic similarities more accurately show our evolutionary relationships. So as time has gone on, we've grouped things more accurately based on the genes. Next thing we're going to talk about today is a thing called cladistics. Cladistics is classification based on common ancestry. And phylogeny is the evolutionary history for a group of species. And this uses evidence from living species, from our fossil record, and from molecular data like DNA. And it's shown with a branching tree diagram, just like that below. This is also going to go along with your assignment number 18, where we're going to look at this a little bit more in depth. Cladistics is a common method to make evolutionary trees. And classification is based on common ancestry. And our species are placed in an order that they descended from some kind of ancestor. Look at this. Cladogram is an evolutionary tree made using cladistics, just like your assignment 18. So a clade is a group of species that shares a common ancestor, and each species in a clade share some traits with that ancestor and also have some traits that have changed over time. If we look at this on our cladogram, we're going to have these things called derived characters. They're traits that are shared in different degrees among all of these different clade members. The clades are like the groups, and it's for the basis for arranging the species on our cladogram. The more closely related the species are, the more shared derived characteristics, okay? So the more things that they have in common, the more closely they're related they are. And they're represented on a cladogram as little hash marks, little lines, okay? If you can look down here below at my actual cladogram on the slide, you're gonna see this thing with 4 limbs, embryos, skull openings, feathers, and toothless beaks. All of these are derived characters. They're basically characteristics. Now, nodes represent the most recent common ancestors of a clade. So I'm going to get to this with my little lines and arrows showing each of these things. So down there, you see that line right there? That's a node. Okay, it represents the most recent common ancestor. Okay. We also have on here the clades, which are the different groupings. And clades can be identified by just snipping a branch right underneath that node. You got a clade, which are all just like one grouping of organisms. And again, molecular evidence reveals species relatedness. Molecular data may confirm classification based on physical similarities, and a lot of the things that Linnaeus did was correct, okay? But we also may propose new classifications based on the data, and in general, DNA is given the last word by the scientists, so we might have to reorder things based on new genetic and DNA evidence based on this classification system. From here, I would like you to take a few minutes. We are going to work through assignment number 18. Okay, so I would like you to pause this, go to assignment number 18 so you can look at the cladogram. I'm going to post another video on that. Actually, you know what? Let me go ahead and do it right here. So we have our assignment number 18. And it's basically showing the cladogram down here below. All right. And basically it says analyze the cladogram. If you read above, it just explains what a cladogram is and some of the vocabulary that we took already in our notes. But basically, examine a sample cladogram. Each letter on the diagram points to a derived character or something different or newer than what was seen in previous groups. Match the letter to its character. Now, this is not a real example, okay? It's not a real life situation. It is showing all these insects on here to be something simplistic for you to look at. So if we look at this, we've got all of these different derived characters written underneath them. Wings, double set of wings, curly antenna, 6 legs, segmented body, crushing mouth parts, and cerci, which are abdominal appendages. And it's your job to determine where they fall on our cladogram. So if we look at number 1, I'm just going to give you a few of them. If you look at number 1, where it says wings, which letter would you place that on the cladogram? So now you've got to look at your cladogram and determine, would I put it A, B, C, D, or so on. So everything after that point, wherever you place it, needs to have wings. So if you look at this, if we look, this organism has wings, this one does, and this one does. But everything in front of that does not. So you would want to put wings down here at letter F. So number 1, F for wings. Then let's look at number 4. Double set of wings. Okay, well, now if we look at everything that has wings, there's only a couple of them that have 2 sets of wings. We would have our butterfly and our dragonfly. So our double set of wings must fall here at G. So 4 is G. Number 7, curly antenna. Where would I put that? Well, if I look, there's only one organism on here that has a curly antenna, and that is over here, the butterfly. So I definitely am going to put curly antenna being H, so 7 would be H. I want you to go ahead and fill in the rest of them, doing just like what we did there. Then what you're going to do is you're going to move on to part 2. Okay, part 2 is going to be making your own cladogram. So to make a cladogram, you must first look at the animals you are studying and establish characteristics that they share and ones that are unique to each group. For the animals on the table, indicate whether the characteristic is present or not. Based on that chart, create a cladogram like the one pictured above. So if you look at this, okay, we need to determine, we've got all these organisms over here. We've got slug, catfish, frog, tiger, and human. And then we have all of these derived characters, cells, backbone, legs, hair, and opposable thumbs. For each group, I want you to put a check mark if they have it. So if we look at a slug is like a snail without a shell. Does a slug have cells? Yep. So you're going to go ahead and put a check mark in that box. Slug has cells. Does a slug have backbone? No. Legs? Nope. Hair? Nope. Thumbs? Nope. So no more check marks. Catfish. We're going to look at catfish next. Does a catfish have cells? Yep. Does it have a backbone? Yep. Legs? No. Hair? No. Opposable thumb? No. Okay, you're going to do that for the rest of them. Okay? All right. The next thing you are going to do then, once you have determined which organisms have which characteristics, you're actually going to draw a cladogram. You're going to do it just like the example in part 1. Now, you do not need to actually draw the animals. You can write their names. You can write slug, catfish, frog, tiger, and human. But the first thing you need to do is to draw a line that looks like that. Just draw a straight little line. Okay? From there, you are going to make branches that represent each of the different organisms. And we have slug, catfish, frog, tiger, and human. So you can see now I've made some branches. Okay. From there, we're going to label each of the branches. So I'm going to put slug, catfish, frog, tiger, and human. So you can see it looks just like that. Hopefully you can see it. Okay. Okay, now we need to determine where all of our derived characters are going to fall. So we're going to have these little nodes down here, which are little tabs or little hash marks. So now I've got my little tabs. You see those down there? Those are going to represent my different derived characters. And my first one that I'm going to put there, what do they all have in common? So if you look back at your chart, what do they all have? Well, hopefully you're saying that they all have cells. So you're going to go ahead and put cells down there. They all have cells. Okay? So everything past this point has cells. Now we need to determine what's going to go here. So what does everything past this point have? Hopefully you look at your chart and you determine, oh, everything past that point has the backbone. So you're going to go ahead and write backbone. Okay? You're going to go ahead and do that for the rest of the derived characters. And then make sure you turn that into me. That is assignment number 18. Okay? From here, we're going to continue with our notes. Okay? So classification is always a work in progress. The new tree of life, now that we use currently, shows it changes. Okay? Down here it details some of those changes. You don't necessarily need to know those changes. Just know that things have happened to change things over time. I'm going to grab something here. Okay. So, moving on. Our modern day taxonomic groups involve a bigger category. Remember in Linnaeus, he started with kingdom? We have one that actually goes above that now, called a domain, okay? Then we have the kingdom, kept the phylum, kept the class, the order, family, genus, and species, all right? The broadest one now is going to be the domain. So basically, everything alive is going to fall into a domain, and there's actually 3 domains. Species, again, is the most specific. Only one type of organism in the world will fall into that category. The 3 domains in the tree of life are bacteria, archaea, and eukarya. And again, domains are above the kingdom level. And they were proposed by a guy named Carl Wohlsey based on RNA studies of prokaryotes. And domain model shows more clearly these prokaryotic divisions because before all the prokaryotes were together, and really that wasn't truthful. And our evidence, our molecular evidence, our RNA has actually shown that. So domains, for you to know, are above the kingdom level. There's 3 of them: bacteria, archaea, and eukarya. Okay, so you are going to grab this sheet now that you should have downloaded this along with our notes. You're going to be using this to gather information for the rest of the slides. Okay, so right now I want you to identify the 3 domains. We've got archaea, bacteria, and eukarya. Okay, so please write those down. Archaea, bacteria, and eukarya. Okay, so then we've got our kingdoms we're going to put under them. So I've made this slide to kind of show you exactly what you're going to put on the sheet. So archaea, bacteria, eukarya. Go ahead and write the kingdoms under here. We have archaebacteria, eubacteria, protista, fungi, plantae, and animalia. Those are the kingdoms that each of these fall into. And you'll notice that archaea only has 1 kingdom, bacteria only has 1 kingdom, but eukarya has 4 different kingdoms. So it's going to involve a lot more organisms. Okay, so I just want to talk about the first 2 domains and their cell types. So archaea, bacteria, and again, you don't need to write anything down at this point. You're going to be filling things in the chart, and I'll show you that as we go. So archaea and bacteria are prokaryotes. They have no nucleus or membrane-bound organelles. They're unicellular, okay? Archaea includes prokaryotes in the kingdom Archaebacteria. Their cell walls contain this thing called peptoglycan, but are chemically different from bacteria, and differences were discovered by studying RNA. They're generally unicellular, and they can be autotrophs, which means that they make their own food, or heterotrophs, which means that they have to ingest or eat food as well. Okay. The domain bacteria includes prokaryotes in the kingdom Eubacteria. It's one of the largest groups on Earth, bacteria. Classified by their shape, need for oxygen, and they also cause some diseases. They also have cell walls with this chemical called peptoglycan. They're all unicellular, meaning they're one cell big, and also they can be autotrophs, making their own food, or heterotrophs, where they ingest other things. So I'm going to have you fill in the first couple of columns here on your set of notes. This is what we just talked about. So basically, we're going to just be talking about the domains Archaea, bacteria. You have your kingdoms, prokaryotes, kingdoms Archaebacteria, Eubacteria. Cell type, you're going to put in prokaryotes for both of them. Cell structures, you're going to go ahead and fill those in. Cell walls with peptoglycan. Number of cells, you're going to put in unicellular. And then mode of nutrition, how do they eat? Well, they can be either autotrophs or heterotrophs. So make sure you go ahead and fill in that chart. Some facts and examples about each of them. And again, I'm going to go ahead and talk about it first, and then we'll add some information to our classification chart. They're probably the first cells to have evolved because they come in very, very, very harsh environments. Remember when we talked about the origins of life? When we talked about those horrible things like pools of lava, explosions, meteorites, boiling seas, all these harsh conditions. Archaebacteria are like ancient types of bacteria that can actually live in these extreme environments. And usually we can find them in sewage treatment plants, thermal or volcanic vents, hot springs, geysers that are very acidic, and in very, 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 very salty water. So they live in extreme environments. Some examples of eubacteria now. Eubacteria, a lot of them, or some of them, cause diseases. Okay, so bacteria, like the things that live on our skin, causing us to get sick. Some can be found in basically all habitats everywhere around us. They're not in the harsh environments like those Archaebacteria, though. They're important as decomposers for the environment because bacteria, you know, break things apart and decompose them. And they're also commercially important for making things like cottage cheese and yogurt, buttermilk, things that are fermented. Some examples are E. coli and then Streptococcus, which is strep throat. Okay. So now we are going to add some facts and examples to our two groupings here. And again, I wasn't expecting you to write the notes in your notebook. I'm expecting you to write it down here on the chart. So go ahead and write in the different facts there on the chart. We're going to move on to the next one now. Now, bacteria and archaea can be difficult to classify, and so at first they were grouped together, but as scientists have learned more, they have gone and put them into different groupings. But that may change over time depending on more information as we find that out. Okay, so next we are going to be looking at our next groups, eukarya. Eukarya for eukaryotes, because they all have eukaryotic cells. We have the kingdoms of Protista, Plantae, Fungi, and Animalia. So again, please make sure now we're over here and looking at our eukarya. Okay, you have your different kingdoms. Cell type, please write in eukaryotes for all of those. Moving on, if we look at Protista, and again, I'm going to talk about it, then we'll add it to our chart. They have cell walls made of cellulose. Some actually have chloroplastic plant cells. Most are unicellular, but some actually will live in colonies where they group together. Some are also multicellular, but most, like I said, are unicellular. Some are autotrophic, and some are heterotrophic. So some make their own food. So the ones that have chloroplasts make their own food, while others do need to ingest things and eat them. Most of them live in the water. They're aquatic. Some examples are amoeba, paramecium, slime molds, giant kelp. And then some are also parasites like malaria. Okay. All right, so I'm going to have you go ahead and fill this in for Protista. Take a moment, fill it in. I'm going to move on to the next one. You can pause it so you can fill it in. Our facts and examples are there. Again, pause it so you can fill it in because I'm going to quickly move to the next one. Then we have fungi. Fungi have cell walls made of chitin, which is like the hard outer like exoskeleton of like an insect is made of chitin, kind of like our fingernails. Those are keratin, but chitin is kind of similar to that. They're multicellular except for yeasts. They are absorptive heterotrophs, meaning they digest food outside their body, okay, and then absorb it. Mushrooms and yeast are some examples. Again, go ahead and fill in your chart. Fill in your facts and examples, and then we'll move on to the next one. The plantae. Plantae have cell walls made of cellulose, which is a fiber. Okay, they have chloroplasts because they need to do photosynthesis. Multicellular, some green algae, however, are unicellular, but most of them are multicellular. Again, they're autotrophic because they make their own food. They do photosynthesis, so they absorb sunlight to make glucose. And things like moss, fern, flowering plants, those are just a few examples. Again, we're going to go ahead and fill in our chart. So go ahead and do that. Again, pause it so you can fill it in. And add your facts. And then we're going to move on to Animalia, which are animals. They have no cell walls. They do not have chloroplasts. They are multicellular. They are ingestive heterotrophs, meaning they have to eat food. They consume it and digest it inside their bodies. They feed on plants or animals. Some examples: sponges, coral, worms, fish, insects, birds, amphibians, reptiles, and mammals are all examples of animals. So go ahead and fill in your chart. Add your facts and examples. Okay, and that is it. Okay, so you have your nice little chart here. You are going to use your chart then to do assignment number 19. So your next thing you're going to do, look at assignment number 19. You'll notice I have our domains, our 3 domains, and then I have our kingdoms over here, which I also have up there on the slide. What you need to do is take a look at your chart and write in 3 facts for each of the domains, about each of those 3 domains, and then 3 facts about each of those kingdoms. Any facts that you have on your chart that you just took, you can go ahead and add it. It could be eukaryotes, it could be eat food, whatever it is. Go ahead and write down 3 facts for each of those groups, and that is your assignment number 19. All right, guys, have a great day.